What is the idea of the technology you're working on, and what do you think is the motivation behind it?
Mhm. Hi, I'm Anna Blakney. I'm an assistant professor here at University of British Columbia in the Michael Smith Labs in the School of Biomedical Engineering. My lab works on RNA vaccines and therapies, and the technology we wanted to share with you today is self-amplifying RNA. So for those of you who have never heard of this before, self-amplifying RNA is just a type of mRNA that is able to make copies of itself once it gets into a cell. So what this means is that we're able to use a much lower dose of RNA and we get protein expression for much longer in vivo.
What do you think is the advantage of this technology?
Hi, so I'm Cynthia, and I'm a graduate student at the Blakney Lab. And my research is on developing gastrointestinal disease therapeutics as well as constructs for gene editing. And I think the big advantage of self-amplifying RNA, since it encodes that replicates, is that it can amplify a lot of your gene of interest. And because of that, we get a lot longer expression and kind of more protein produced than mRNA. And because of that, we can also use a lower dose. So another advantage to that is that you can reduce kind of those dose-related toxicities. And I think because of that, it also is an advantage for kind of scaling it up for manufacturing as well, since you can use lower dosages per person.
Hi, Nathan. So, what was your biggest challenge you encountered when you developing this technology?
I'm Nitin Battala, a fourth year PhD student here at UBC under School of Biomedical Engineering. So I work on self-amplifying RNA to make therapeutic antibodies out of it. So the biggest challenge in this field is like the self-amplifying RNA itself. So self-amplifying RNA is like a couple of folds longer than mRNA, so which makes it more prone for degradation, which we put a lot of efforts to make sure we work intact RNA as possible. Yeah.
What are the potential shortcomings of the technology, and how are you looking to further improve this technology in the future?
Hi, I'm Kay. I'm a graduate student at the Blakney Lab, and I'm working on applying saRNA or mRNA research for cell reprogramming. So one of the potential shortcomings that we have in our research is immunogenicity or reactogenicity that we get from delivering RNA to tissues. So some way we could fix that is by making the RNA or the LNPs very cell-specific or tissue-specific, so we can modify lipids or we can modify the RNA itself so that it can be more specific and minimize adverse effects.
All right, Jackie. So beyond the initial motivation, where do you see this technology in future terms of further applications?
Hi, I'm Jackie, and I'm the research technician and lab manager in Blakney Lab. I think self-amplifying RNA already have like approved vaccine already on the market. So I think self-amplifying RNA can also be really useful for gene editing, delivery, and protein replacement, even just like cancer biology or like cancer treatment. So by delivering lower dose of RNA treatment to animals, we can get a better and longer lasting protein expression. So that's one of the advantages of this technology. I think siRNA has a really big and bright future.
Hi, everyone. Welcome to our lab. Today, we're going to show you how to make an saRNA formulation and test it in cells. Come on in. The first step is to make DNA, and we make that from our bacteria. So we're going to choose one of these little colonies, take all the DNA out of it, and purify it before we make RNA. Let's get started.
All right, now that we've got the DNA, we will make the RNA using in vitro transcription, where we have NTPs, the nucleotides, we have the enzyme, the T7 enzyme, the buffer, and some water. So we'll mix all of these components and we will have our RNA. All right. All right, so now we have our RNA and then we have to do the quality control to measure all the properties of the RNA that we've made.
As we have the RNA, let's check the integrity of it now. It looks like a really good quality for RNA.
Right. Now that we have our RNA here at the LNP room, we're going to make our lipid nanoparticles. So I have my lipids here, and then we're going to attach a syringe to the other side for the RNA, and we're going to use this machine to mix them together.
Now we have the LNP lipid nanoparticle that's characterizing. So this is a machine it's called DLS. We want to check the size of the lipid nanoparticle and the zeta potential, which is the surface charge of the particle. Let's do it.
So now that we've characterized the LNPs, we're ready to test this formulation in cells. So this can be done in hex cells, which is what we commonly do, but you can also do different types of immune cells or primary cells. So in this case, what we're checking for is replication or protein expression and their amplification kinetics. So in this case, I will take up a dose of the RNA and then transfect it into my hex cells. And then now we can check for fluorescence.
All right, well now we know that the saRNA works, it's time to test it in vivo in mice in a therapeutic model. Thanks so much for joining us today and hope to catch you next time. Bye.